Right, here we go with our class. And let's see, okay. So this week, where are we at? And we're doing fine. And remember, we have Riley, and Riley's going to come again pretty soon and talk to us about the receptor for that. Then we're doing chapter 9. Chapter 9 is about transcription regulation, and we will cover that this week. And so then we want to start moving on to chapter 17, which we have been foreshadowing since day one. So that's about receptors and signal transduction and all about that. And so we will definitely be completely dealing with chapter 17 next week. And so that's where we're at. And then here's this example of a question. So Joe sent me a screenshot of this, and you'd have to scramble to write this down because it's from chapter 9, which we'll deal with this week. And here's just an example of our book writing, and our book is just like, so what do you have to know about that? And the answer is going to be, we need to know like 2 points worth of stuff about transcription elongation. And there's a good chance we'll get to answering that in that context today, or if not, definitely Thursday. And so, we see that our book is just stacked up with more details, and a key thing is to sift through those and try to build sort of a hierarchy or a pyramid of knowledge. And I think that's a center part of being a biologist, is by biology has so many examples, and we have to sift through them and organize them and not necessarily remember all of them, but remember the most important ones first on down the list. Okay, so good to ask questions, good to send me questions and things like that. Okay, so we are going to follow up on this. Another thing from Joe, and that is, I think in the concept chat at the end of class last week, we were talking about MIC. And we're going to go back and make sure we understand MIC, because there was a confusion. So MIC we talked about in the context of oncogenic retroviruses, and then I started talking about it in the context of translocation, chromosomal translocation. So we are going to just go back and talk about kind of everything we need to know about MIC right now, just to make sure we clarify that, okay? And so MIC is an oncogene like that, okay? And so let's try to also kind of back up and sweep through those examples again, like that. So here's this list of these oncogenic retroviruses that we're going to need to know about. So remember, when we think oncogenic retroviruses, we think 1970s. And just getting a grab of about 30 oncogenes that are in our bodies and orthologs in all other animals, or lots of other animals. And they can get mutated and cause cancer, and we find them in cancer, right? Like that. So we'll eventually talk about this many of them. These will be good examples of those. I added one here, KIT, that wasn't on our previous list. And here's a key thing right here. Of these, we're going to learn about RAS and MIC for our first exam. These others we're not going to learn about until chapter 20. But I just wanted to show you that, and so we can point to that whenever we want to, right? Back in the 1970s, they got a hold of some of these, and they know a lot about them now, like that. And they were found in these oncogenic retroviruses. They generally have these 3-letter names, like that, right? Okay, so let me just show you this, okay? So we have our signaling pathway for normal wound healing. Remember, we have an example of a chromosomal translocation where the woman got skin cancer because she accidentally rearranged her chromosomes to bring the regulatory region of skin collagen upstream of the transcription unit for PDGF. And suddenly, she was misexpressing PDGF in her skin cells. And that just created a self-stimulatory thing. There was no wound to heal. Her body was just stimulating this pathway. And this pathway includes RAS. So on the oncogenic retroviruses, scientists have been learning about RAS since the 1970s, and our author is one of them, okay? Then these 3, RAS, ERB, and KIT, these 3 are all receptor tyrosine kinases. In our body, there are 58 of these, and we're going to learn about these 3 and maybe one more, like that. So that's what those are. They'll fit in. Now, they fit in here, and they have different signaling molecules that bind to them, right? PDGF gets activated by the PDGF receptor, okay? So they get activated by different molecules, and then they do have some different downstream signal transduction pathways, and definitely different cell cycle or cell responses, like that. So we're going to learn more about not just RAS, but some other ones. But just to show you on this list, it's a pretty easy list because the first three are examples of the receptor tyrosine kinases, and we're going to be really familiar with the PDGF receptor. So we can plug these stories in. Oh, sorry, not the first three. RAS is not one. These two are receptor tyrosine kinases. I apologize for messing up. These two are. Then we learned about RAS, and RAS is on this pathway, so that's where RAS fits in. And then we'll talk just a little bit in our course about SARC, and we'll learn in chapter 20 there's a SARC homology domain. So SARC fits in this pathway. And then June and FOS and MYC are all transcription factors. And we'll learn that MYC, like ELK, is one of the downstream targets of this kind of signaling pathway. And in a different pathway, June and FOS are activated. So this pathway is kind of one of these universal drawings for us that we go to in our mind. And we'll see when we talk about chapter 17, we will draw that out and be like here and here and here is where these fit in, okay? So just to give you a little overview, and now we're going to dive into RAS and MYC. What do we need to know about RAS and MYC? Well, there's something we know about RAS, okay? So let's just see if people can list some things that we have learned about RAS. What have we learned about RAS in our course? Statements that you could make on an exam about RAS. What would they be?
It's a part of a lot of signaling pathways.
Yep, so we learned about, so it's part of a lot of signaling pathways. And it is specifically on this kind of a pathway. It fits into this pathway. And this pathway is seen in normal skin healing. It's seen in that woman with cancer. And then it's a lot of other growth pathways in our body, right? So yeah, it's a signaling molecule in these pathways that look like that. Yep?
It's mutated in a lot of cancers.
Yep. So RAS is a protein, and that means it has to have a gene, and that gene can get mutated in and it does. It's the most commonly mutated gene in human cancers. And it really typically has point mutations in the coding region. So we make a different protein. Yep, so that's another one we learned about RAS. What's it? I think Hannah had her hand up.
Yeah, it either binds to GDP or GTP.
Yep, so it's this monomeric G protein, and it is always bound to either GDP, inactive, or GTP, active. Yep, what's another thing we learned about RAS?
When it's unable to, is it hydrolyzed GDP? That's when it becomes cancerous?
Yep. So all of the mutations of RAS have the same problem, and that is they are point mutations in the coding region, and we make a different RAS product. There's not the regulatory region of the gene, they're in the coding region, and we make a different RAS product. And the product we make is always the same problem, and that is that when RAS is active, it's bound to GTP, and it inactivates itself by being an enzyme that breaks GTP down to GDP. It hydrolyzes it. And so it inactivates itself. And all of the mutations in RAS that are in cancer are mutations that change amino acids that are in the enzymatic domain, the catalytic site, the active site, that is going to catalyze the breakdown. And so the problem with RAS in cancers always is it fails to turn itself off very well. Different mutations, sometimes it turns it off slowly, sometimes not at all like that. So it's just not turning that pathway off, and that pathway stimulates growth. So it's activated like that. Okay? Yep.
And that's just from the wrong amino acid being the enzymatic site.
Yes, yeah, so in an enzyme, it's a protein, it has a region called the active site, or the catalytic domain, and that's what's going to bind the substrate, in this case GTP. And so there are going to be amino acids lining this domain, and those are the ones that have mutations and make the wrong amino acid, so the enzyme just doesn't function very well and turn itself off. Let's see, yeah, that's great. And maybe there's one more thing we could, or maybe there's more things we could say about RAS. What's something else we learned about RAS? Yep?
PDGF helps turn it on and something.
Yep, it's right there, it's on this pathway. So our specific example is that wound healing pathway, that natural pathway, and under a wound, PDGF is activating the PGF receptor and RAS and this pathway, like that. So we get ELK activated as a target, and that's all happening under a wound in a skin cell to help the skin cell hurry up and divide and grow and heal. Another thing is RAS was first discovered as an oncogenic retrovirus. So that's something you could mention about RAS that we know is that it was one of these really early ones, and then scientists, found that it was mutated not just in that, virus infections, but in natural, mutations. Yep. And that's a pretty good list. We know a lot about RAS. Now we're going to do the same thing with MYC. Okay. So there's RAS, and here's MYC. And so this is what we want to learn about MYC. So MYC is a transcription factor. And so we are already experts. We know what that means. It means that it's a gene, and it's going to get transcribed similar to MyoD and get synthesized as a protein, MYC. And it's going to bind to target genes. There'll be lots of MYC molecules made, so it can activate lots of target genes, and it does. Yep. And later in the cell cycle chapter, we'll learn what its targets are, and it's the targets that activate the cell cycle, that help the cell go through the cell cycle. So we'll learn more specifically about its targets, but it stimulates the cell cycle like that. And let's see, Hannah, did you have a question about that, or that was sort of about it? Okay, so it's a transcription factor. So we could just get out clay right now and make that model of seeing MYC get activated like that, okay? So how does it get activated? And one way is this pathway. It gets activated a bunch of ways, but we learned this pathway activates ELK. It also activates MYC. And these activations vary in different cells and so forth, like that. Where is an example where MYC is normally used to stimulate growth? And the answer is in our bone marrow. And so let me, I'm going to have some of these. Yep. So just some little highlights, not from our book, just from a review. So MYC is a global transcription factor. That means it has lots of targets. It turns on lots of genes, kind of like myogenin. Myogenin turns on a lot of muscle genes. These aren't going to be turned on muscle genes. What kind of genes are they going to turn on? Genes that will stimulate the cell to grow and divide. Okay? Where's an example of that normally happens in our bone marrow when we're making new blood cells every day of our life? It says the bone marrow of adult humans makes 300 million cells every minute of our life. So we have a lot of cell division in our bone marrow, growing really fast all the time. And MYC is normally involved. MYC gets activated, right? In that environment, MYC gets activated, stimulating this go, go, go. That has to be controlled, really carefully controlled, right? So that's an example, right? MYC, it's transcription factor. And how's it get activated? Well, that's one way it gets activated is by that pathway, like that, okay? Then, what's it normally do, right? And then how can we have problems with it, okay? So, cell cycle. When we think of MYC, cell cycle. And it does other stuff too, but cell cycle for us. And then it includes this RAS, RAF, MEC, ERK pathway. So that can activate MYC. So that's a go-to. There are some other pathways that activate MYC, but that's one way of normally activating MYC. So then the next thing is that MYC is involved in cancers. It's especially involved in cancers of the blood, leukemia, and lymphoma. I think that's not surprising given its normal healthy role, stimulating the cell cycle in bone marrow. And so it's set up to play a role in our blood cells, and it can get out of control. So that's just that. Okay? So it's regulated. So now we're just going to learn a little bit about how it's normally regulated. And the answer is that when MIC is active, it's dangerous. And so there is a lot of regulation of MIC, a lot of different ways. And they all revolve around, let's just not make it. Or if we make it, let's get rid of it really fast. So that's the plan, right? So we're just going to go through some of those. So it's really closely, tightly regulated, okay? So, let's see, I got these a little bit out of order. But here's the first thing. So we have our list of transcription factors. And MIC is regulated by mRNA stability. So the gene gets expressed, and we have the MIC mRNA, and it just gets degraded really fast. So it's not stable. We're not going to make much MIC. We're just going to degrade it fast. So we can add that to this list. And I guess I sort of have messed this up because we haven't gotten to this one yet. Oh no, we do. Let me just back up before mRNA stability, because I was trying to get this. Yeah, I missed this. So let's just go through a list. How do we regulate MIC? So the first one is we regulate the transcription of it. And so that's going to be a chapter 9 topic, and we'll come back and talk about this when we talk about this. It goes to that question Joe had from the book. What is this transcription elongation? For us, for the moment, just know that here's the MIC gene, an RNA polymerase lands and binds and starts to transcribe it, and then just stops. Because it's like, we shouldn't be making this gene. It's a big mistake. So let's just stop and wait, and let's hear from somebody else to see if we really want to make this. So that's the first thing. And we can add that to this list, right? So these are transcription factors that we're learning about and how they're switched on and off. And one way to regulate MIC is to control how well the RNA polymerase can transcribe it. So that's adjusted. And normally it can't get it done, but when we activate the pathway, it gets a little bit more stable, like that. So that's the first thing, or just a little more elongation. So the second one is mRNA stability that I jumped ahead to. The mRNA isn't stable. It's just going to get degraded. So we don't want to make it, we don't want to transcribe it, we don't want to keep it around. And so we have an example of that. mRNA stability is a way to regulate transcription factors. Just make the RNA stable or less stable, and we can control it. Then another is we don't want to translate the mRNA. Let's not even make this RNA. Yep?
What do you mean by make the mRNA more stable or less stable?
Yeah, so I mentioned before that when genes are expressed, they're constantly expressed because RNA is unstable. And it just gets broken down, it gets degraded, it has a half-life. And that can be regulated. It can vary. So we could break it down fast and turn the gene off. Or we could stabilize it and let some more be made. Yep. Okay. Then here's the next one, translation. So yeah, even if you can get the mRNA to the ribosome, let's not translate it into a protein. Let's control it at that level. So we add that to our list. You could regulate translation and just say, let's not make it. Even if the RNA is there, let's not make it. Okay? And then the last one is, even after you make it, the protein's unstable. It only lasts for about 10 or 20 minutes and it's gone. So again, all those collectively fit into, let's just not have it around. You know, you want to transcribe it, I want to pause. You have transcribed it, I want to break the RNA down. You have the RNA, I don't want to translate it into a protein. You have the protein translated, I want to break that down.
Just at every stop, it's just like
Every possible thing. You're like, no, no, no. And it goes back to this example of like, oh, you have a gun in your home and you have kids at home. Well, you better have the gun unloaded, hide the bullets, lock the drawer with the bullets in, hide that key, put the gun, safety on, unloaded, in a cabinet, lock the cabinet, hide the key. That's MIC. MIC is kids at home with guns. You know, you're just like, somebody's going to get shot. It's super dangerous, right? And where is it being used? Normally in our bone marrow, playing with fire, right? It's a dangerous game to simulate the cell cycle in an adult. So that's it, okay? So then what happens in cancer? So, okay, so that's its normal thing, and it's just extremely regulated, like that. And that's what we want to think about. We want to think about MYC as just being a dangerous protein, and it can activate a bunch of genes and send its cell into the cell cycle, and we want to be really careful about that. It happens in our normal body, but we want to have it tightly regulated. So the next thing is in cancers, MYC does not have mutations in the coding region. Remember, we learned about RAS. So I just want to go back to this and go, in comparing RAS and MYC, RAS has mutations in the coding region, and we make the wrong protein. MYC makes the normal protein, and the mutations are in the regulatory region, and we make too much. So we overwhelm those protection mechanisms. We have so much MYC activity that the elongation happens, we get some RNA, and then we make so much RNA that it's stable enough, and then there's enough of it that some gets trapped. So we just force express, and that's called overexpress, too much. And that's not wrong product, that's not a coding region problem, it's a regulatory region problem. So that's always the case with MYC, is they have a wrong regulatory region, and we're just making too much MYC. And it just overwhelms those. You can do that, right? You can have like a dam, and then here comes a bunch of rain, and the dam just gets overwhelmed, right? Whatever protective mechanism is overwhelmed by overexpression, and that's what it's called. We just overexpress normal MYC, and it's too much, and we get some, to activate the cell cycle, okay? So that's a good compare and contrast with RAS and MYC, right? Compare the mutations of RAS with the mutations in MYC that are seen in cancer. 2-point question on the exam, and you say RAS mutations are in the coding region, and we get a defective protein. MYC mutations are in the regulatory region, and we get too much MYC. See, like that? So RAS, MYC, like that. Yep. Okay? All right. So then, that's that. So then, this overexpression is going to be, when you say overexpression, right, that means regulation of the transcription, right, the regulatory region. Okay? So here are 3 examples of that happening to MYC in cancer. So the first one is that they found it in oncogenic retroviruses. And I mentioned before that the MYC in the retrovirus is normal, but it's near the virus gene regulatory regions. So let's just draw a picture of what happens to MYC, because we can draw it or we can make it in clay, right? So here's the MYC gene, and there's the transcription unit, right, that's going to get transcribed into RNA and the output, and then here's that regulatory region. And here's what always happens in cancer, is we still have the normal MYC, and we somehow have the wrong regulatory region. And this regulatory region is what people call a strong region, where we can make lots of product. We're just going to make a lot, okay, like that, okay? All right, and so the first one is, this could be in the retrovirus. So in the retrovirus, that's what we have. We have the retrovirus and a little part of MYC, and it's just the transcription unit of MYC. And the retrovirus has strong regulatory regions, because when it's infecting itself, each one makes a lot. I want to make a lot of this virus right now. So it has, viruses always have some of the strongest regulatory region of any genes, because they're like, we're taking over, and this is how we're going to do it. We're just going to make a huge amount of our stuff. And that's exactly what happens when it's in the retrovirus. Remember when RAS got picked up by a retrovirus, it was a mutated form of RAS, defective form, out of control. This is normal MYC protein, but just along with that infection. So that's the first one, and it makes sense. Makes sense about our clay model of genes, right? Is, we're just going to make too much. We're going to overexpress MYC by using a different regulatory region. Here's the second one, and this is where we get to that confusion at the end of last Thursday, right? So Joe says, let me just understand these. Two different stories. So the first one is that oncogenic retrovirus story, and it's just because there's too much MIC because the retrovirus has these really strong regulatory region and makes too much MIC. Just too much MIC. It's normal MIC, but it just overwhelms all of those checks and balances, right? Like that, okay? So then the second one, totally different example. Same thoughts and ideas, and that is this chromosomal translocation. So that's different. The retrovirus was just in the 1970s, it wasn't found in a human, like that, okay? Here, chromosomal translocation. Now, we just have to mention that, remember, the woman who has skin cancer, she has a chromosomal translocation. She's translocated the chromosome to bring the regulatory region of skin collagen to the transcription unit of PDGF. So two different chromosomes got torn and glued back together, and it's an error. Okay, misexpression, that's called. In this case, we're going to look at one example of chromosomal translocation. These are in humans, right? So a totally different story than the retrovirus story. It's a human, and it's a human walking around, and they got cancer. And the way they got cancer is through a chromosomal translocation, and the example we're going to talk about is an example called Burkitt's lymphoma. So there are some cancers of our blood and lymph system, and so they tend to be these circulating immune system cells that have somehow gotten out of control. And we learned that normally MIC is helping regulate the production of blood cells in our bone marrow. So it's not too surprising to see there could be a chromosomal translocation where we just get MIC going in those blood cells. Too much, like that, okay? So here's this Burkitt's lymphoma, and so Burkitt's lymphoma, and it's a translocation of MIC, like that, okay? And it says all types of Burkitt lymphoma are characterized by that. So this Burkitt's lymphoma is a fairly common kind of lymphoma, and everybody that has it has a big problem with MIC and overexpression of MIC, right? And so go to this drawing, and what's happening here, okay? So in our body, in our blood cells, we have three major kind of blood cells that are constantly being made by our bone marrow. So we have our red blood cells, and then we have our white blood cells, and there are really two main types of those. One is called a B cell, and one is called T cell. Okay? So this isn't a course in immunology, but just to say a moment about this. So red blood cells, they carry oxygen, they have hemoglobin, great. Then white blood cells, they're involved in the immune response. Riley is going to talk to us about T cells, and T cells are helping identify foreign things and helping kill them. B cells also help. B cells are the cells in our body that make antibodies, and with COVID we've heard a lot about antibodies. Antibodies are proteins that help stick to foreign molecules in our body. Antibodies are proteins. So in the B cell, there's are genes that are antibody genes, because antibodies are protein. And so here's an antibody gene, and it has a transcription unit, and it's going to make RNA and get translated to antibody protein. And that's what this B cell is supposed to do, and it makes a lot of antibodies. That's its special role, is to make a lot of antibodies. And make them fast, because it's all about infections and fighting infections, so let's fight this infection. And so, because of that, this regulatory region is really strong. And what happens in Burkitt's lymphoma is a translocation that brings the antibody regulatory region, just by accident in trying to repair damaged DNA, and it connects it to the transcription unit of MYC. So the translocation in the woman's case, with skin cancer, we make PDGF in the wrong place. We make it in the skin. And people call that, we say, we misexpress it. We made it in the wrong place. In this case, what happens is we just make lots of MYC. And so we say that we overexpress MYC. And that's a translocation that leads to this Burkitt's lymphoma, is you're just making too much MYC. Okay, so that's a different example than the oncogenic retrovirus, right? The oncogenic retrovirus had to do with getting connected in the virus, and this is about getting one of these kind of mutations that's called a chromosomal translocation. DNA gets damaged, it gets repaired, and it just made a mistake and brought two pieces of chromosome together that are wrong. And what we've done is we brought the regulatory region of the antibody gene, connected it to the transcription unit of the MYC. And we're just going to overexpress MYC in these B cells specifically. Why? Well, because there are transcription factors that bind to the regulatory region of the antibody. Those protein transcription factors are active in the B cells, and they're like, go, go, go. So that's what's happening in that example. And that, we're sweeping it back around this to clarify that confusion. We got oncogenic retrovirus story for MYC. That makes sense. Then we have this chromosomal translocation story for MYC, and one example is Burkitt's lymphoma, so we can put a name on one of those. There are other examples too. Yes, Zach?
Just to clarify, I was going to say, so it's using the regulatory region of the antibody, but the coding region of the MYC.
Mm-hmm.
That's the...
Yep. Or we'd say the transcription unit, like that, the part of the gene that's going to get expressed in output, like that. So that's really sad situation. That's what's happening with these people who have Burkitt lymphoma, is they're just making a huge amount of normal MYC, and it's too much to, to predict and limit it with, elongation and RNA stability and translation regulation and post-translational protein stability. It just we get too much MYC getting made for the system. Then the third one is something we haven't talked about, and it's just got a nice name. It's just called gene amplification. And I'll just show you an article to put a context in that. So it says, here's an article that says MYC, gene amplification, is often acquired in lethal distant breast cancer metastases of unamplified primary tumors. So in certain cancers, MYC is a problem, and in those cancers, the normal MYC gene has accidentally been copied over and over again. So instead of just having the normal 2 copies of MYC, one for mom, one for dad, the patient could have 150 copies of MYC. And that's an example that happens in some cancers where they progress, and so cancers progress, they get worse. And the way they get worse is the cancer is growing, and as it grows, it picks up more problems. And so one of these things in a late stage cancer is the cancer cells are just very defective, and they pick up more problems. And one thing they start to do is they just start copying wrong, making mistakes, and they can end up, some of them, just copying MYC.
Does it metastasize because it's blood? Is that why it metastasizes? Like,
What?
does it metastasize because it's like in your bone or like blood? Or is
No, so see, in this case, we're talking about a cancer that was a solid cancer of the breast, and it's metastasizing, it's spreading like that. And so that's pretty common in cancers, that part of the progression is the cancer is localized to a certain place, and then it's growing, and so it's growing faster, and now it's spreading, right? So there are these, these steps, and they involve like getting more problems. We start out with a little problem, we have a problem. Like there are benign cancers like some moles, right? You're like, that's a weird thing growing a little bit weird, we just need to keep an eye on it, because it could pick up another mutation and another mutation and then start to have the same thing. Same thing with polyps in our intestine. Those are like some growth, it's a polyp, it's been some extra growth, shouldn't be happening, but it's okay, not growing fast, but it could pick up another mutation, another problem. And so something that happens in certain cancers is chromosomal amplification, or a gene amplification of MYC. And there, it's just the normal MYC gene, but you just have so many of them that you again end up making too much normal MYC, like that. So and that should just be satisfying with us too, right? Because you go get your clay out and you'd be like, here's the oncogenic retrovirus, here's MYC, and here's the clay piece that's the retrovirus, and it's going to make too much MYC. Here's the chromosome translocation. Well, we have the antibody gene here, and we get it torn, and then we get the regulatory region glued on to the clay piece that's transcripted in MYC, too much MYC. Here's the last one. I made a clay model of this gene, and then I made 150 more clay models of the same gene, and we're in trouble because we don't want to make a lot of this gene, but we have a lot of these genes now, and they're just going to make a bunch, too much, and overwhelm that thing of trying to keep MYC in check. So those are our three, and they should just be now seen as good, independent, separate stories, right? Oncogenic retrovirus, just back in the 70s, that's what was going on there. Then chromosomal translocation. People get Burkitt's lymphoma, that's their problem, like that. Then gene amplification in certain cancers, just get a lot of MYC going into background, just like, whoa, got a lot of MYC there, you got a lot of gene amplification. So those are three good examples of things we understand based on gene organization and how it could go wrong. Yep, let's see, Ellie has a question.
For MYC, when it just has its own regulatory region, is it also strong, or is it only a strong regulatory region when it's paired with something that's going to create cancer?
Yeah, so MYC has got a weak regulatory region. We don't want to make very much of it.
Okay.
And so then when we get the oncogenic retrovirus, we have a strong regulatory region. Now we make a lot because of that. Or we get a chromosomal translocation. Now we make a lot of MYC, like that. Yep. Yep. And even though it has a weak regulatory region, if we amplified it a bunch, we'd have a lot making a little, and that just adds up, like that, right? If you have 150 genes of MYC, you make 150 times more frequently, given its same weak regulatory region. Like that, yeah. Other questions people have? And you can see how these can get organized into these 2-point questions, right? Like that, a couple of 2-point questions like that about MYC compared to RAS and the mutations in MYC and RAS. Compare chromosomal translocation with gene amplification, like that. Ask about questions about MYC as a transcription factor, its role. How are some ways that MYC is regulated? Well, elongation, which we'll learn about, RNA stability, translation regulation, post-translational regulation based on protein stability, like that. Boom, 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 like that. So MYC is a super famous oncogene. It's just a really, really famous one in cancer biology. And it fits in really well with our understanding of molecular biology and genes, gene expression. All right. So other questions people have? Like that. And where we were at in class last time. So I just inserted that in, to clarify that. But also, let's be solid on MYC. Let's be solid on RAS, like that. They're really important genes in our body, then we were talking about this, and this is sort of chapter 4 talk. The science is physical, it's reductionist, use model organisms because of the orthologs, research done in lab, really controlled environments, and then something we've talked about is discoveries get turned into tools, like that. And the book loves to talk about that, molecular biologists love to talk about that. Several of the Nobel Prizes that have been awarded have been because somebody came up with a way of using this as these natural processes as tools to learn more. And so here are 2 examples that we can learn about. One we talked through last Thursday, it's this GFP. That shows up in our first video. These guys won a Nobel Prize. The gene is from a jellyfish. You take the transcription unit from the gene from the jellyfish, add a regulatory region to a gene like insulin, and put that transgene into a mouse, and now the mouse's cells will glow whenever they make insulin. So it's a reporter gene. We talked about that concept, and it came up in our first videos. As we can see here that in the early embryo, this mouse embryo has all the genes, but the genes don't glow. It's the protein products that glow, and the genes are not being expressed. But as cells start to differentiate in the fetus, then these get activated. and we get activation and glowing red cells in the muscle because they probably hooked the GFP transcription unit up to the myogenin regulatory region, and it's just reporting when myogenin gets expressed. So that's one of these examples where the natural biology was just, oh, look at that jellyfish that glows. And then the marine biologist said, I wonder how it glows. What's the biochemical mechanism for that glowing? And he's like, oh my gosh, it's just a protein that's glowing. That's interesting because if you made that protein, then it would be glowing. But the gene doesn't glow, so we could use that as a way of detecting gene expression. Okay? So then our second example is this enzyme reverse transcriptase. And the trick here is it's useful to help study mRNA. So reverse transcriptase is this guy in Wisconsin studying the, do I not have the slide? The replication cycle for retroviruses. So we learned about reverse transcriptase there. The retrovirus has to provide the protein enzyme, and it's going to transcribe the retrovirus RNA into DNA. So that's the enzymatic activity. It's a it's an RNA-dependent DNA polymerase. It uses RNA to guide it to make DNA. And so that's just that, great, good thing, okay? Here's one way that reverse transcriptase is used, and we used it in the laboratory class for this course, and that is this. Something we haven't talked about is that when the RNA is first transcribed, the RNAs in our body typically have regions of exons and introns, and the intron material is going to get removed by RNA splicing. So really typically the transcription unit for our genes is pretty long because they have unnecessary information in these introns. And so it's frustrating to study the transcription unit of a gene because of these long introns. But as the RNA gets processed in the nucleus, the introns get removed. So this is a good visual of seeing that that's really what we want to study, because the red there is the coding region. And you can see that it's all joined together and great like that.
This is during transcription.
Mm-hmm. Yeah, after transcription, this RNA splicing occurs and we remove those introns. And it's, we don't need to deal with it too much in our course because it's kind of an odd detail. But you can see that's frustrating because look, the exons are all scattered around, and those the red is the coding region. And we're like, ooh, that's hard to study. We'd like them all together. Keep it simple. Like, I don't want to study that intron garbage. So what we could do is we could purify RNA, and we could use reverse transcriptase to copy this and study this. It's really hard to study the RNA. It's really easy to study DNA. Reverse transcriptase allows us to copy this and study this. And that's it. Yep?
Do we use a virus to do that?
So that's a good question. The reverse transcriptase is a protein, so the virus must have a reverse transcriptase gene. So instead of using the virus, we can just get the DNA in the test tube, take the transcription unit for the reverse transcriptase, and then add a regulatory region like from a bacteria.
No.
Put that transgene in bacteria, and it will make the enzyme for us, and we can just isolate it and sell it because scientists like to do these kind of experiments a lot. And that DNA that gets made is called cDNA, and it's a copy of the RNA. And people really like to study the RNA for a variety of reasons, and so this reverse transcriptase is used in research labs a lot. Again, left the viruses way behind. They were the source of it, but if we left those behind, we can just get that product and buy it from the store. Before I came here in Seattle, the university was big enough that they had these enzymes for sale in vending machines in the hallway. And you just go up with a purchasing card, and they had several vending machines with different enzymes in them, and you just be like, yep, next to that Snickers is some reverse transcriptase, I will buy that. Yep, yep, yeah, that's just yeah, because it's just a powerful tool. So that's just our second example, right? So kind of this, we'll bring up some other examples, but using GFP is a powerful tool. Then this reverse transcriptase is a powerful tool. We learned about the replication cycle. It's in that, pathway. right? They found it like in the 1970s. The guy won a Nobel Prize, partly because of the oncogenic retroviruses, partly because scientists realized, whoa, that reverse transcriptase is going to be really helpful. Okay, so that's just that, and that's the end of that, because the next thing I want to talk about is chapter 9. Okay, so that's kind of chapter 4 stuff, talking about how the fundamentals of molecular biology involve using discoveries of natural biological processes to create new methods and new tools, like that. Our book, every once in a while, has a box that talks about a method. And to learn 2 points worth of that method, a really good thing is, well, what's that method used for? And then what's the sort of central trick of the method, or tool, feature of the method, like that. So reverse transcriptase is used to make cDNA, right? So it's used to help study RNA, and the feature is that it's this retroviral enzyme that can copy RNA into DNA. So that's the feature of it. Yeah, like that. Okay. So questions about anything at all? All right. So I'll just show you this chapter 9, because chapter 9 is about transcription regulation. So we have our gene in the clay model videos, and so we know a lot of this. And we want to navigate through this chapter and add some more knowledge, but not be overwhelmed, like the screenshot that Joe sent me about transcription elongation, where it's just like blah, 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 blah. Okay? So here's a good way that we're going to add knowledge. Okay? And I'll just show it to you. So we learned about transcription factors like MyoD, myogenin, MYC, ALK, FOXO, and they're going to regulate transcription. And they're going to do that somehow, right? They do something. And there are really 3 kind of most famous examples of how a transcription factor can regulate the transcription. And I'll just say them, and then I'll explain them. But one is they help assemble something called the pre-initiation complex. Or 2 is they remodel chromatin. Or 3, they release this paused or arrested RNA polymerase. So I understand that that's just a list and you don't know anything about that. So that's the list though, just 3 things that are going to be a big piece we add. All right, so now what are we talking about? And we're talking about that clay model video, and so we could use the myogenin gene, and that's the DNA, and it's got an enhancer up here, and the MyoD protein is going to bind. And it's going to activate transcription of the myogenin gene. And so here are the 3 most common ways that it does that. Right? And I don't know what MyoD does, but it probably does one of these. So the first one is that help us assemble the pre-initiation complex. So the pre-initiation complex is here, and we've got our promoter here. And there are several transcription factors that bind to the promoter, and they're pretty boring, but they got to be there. And so you could just make some different shapes like that. So those are transcription factors that are needed, and we need to get them there. And sometimes they're not there, and so sometimes the transcription factor is helping get those. Remember how it's like tethered and it's flopping around, and it could bind to one and help deliver it to the promoter. The other part of the pre-initiation complex is the RNA polymerase enzyme. People would read it like that. Altogether, that's the pre-initiation complex. And RNA polymerase actually has several molecules, like 14, that help it get its job done. like that, okay? So the pre-initiation complex is a whole bunch of proteins, and it has to get piled up there to get things ready to go. And so sometimes the transcription factor helps with that.
This is all happening in the promoter.
Yeah. And it's floppy, right? So it could bind something and kind of keep it in the area until it sticks it down there.
What did you say that complex was called?
Pre-initiation. And that means before we start to transcribe. So when we initiate transcription, when the RNA polymerase really starts copying, then it's post-initiation. So pre-initiation is we just got to get everything assembled, right?
And you're saying that the transcription factors can help bring other transcription factors to that?
Yes,
Okay.
yes. And they help bring these more boring ones that are needed to just get everything organized right here at the start. Like that. All right? So there could be several different ones that are, that one's missing, right? We need to get the gray one. We don't have the gray one here. And that transcription factor's job is specifically bring the gray one. Get that gray one here. Okay, so now when we're talking about that, see what's going on is that the myogenin gene was not being expressed, and it was because something was missing. Just not getting started, or it wasn't coming fast enough. Maybe the gray one would show up every once in a while, but we need to get it there faster, more often, to increase the rate of transcription. So, in the context of this, we had to talk about something called the rate limiting step. And so, I'm not going to quiz you on it, but you would have talked about that in chemistry class. And so, raise your hand if you say, oh, I recognize that. I've heard the rate limiting step. Okay? So, what's happening here is the transcription factor is affecting the rate limiting step. The rate limiting step is that gray transcription factor is not here. So let's get it. And if we get the rate limiting step, we will increase the rate. What we're saying is that there's a rate limiting step in gene expression, and the transcription factor is affecting it. So that's a general thing about transcription factors. You could say transcription factors regulate transcription by targeting the rate limiting step. So the rate limiting step could be assemble the pre-initiation complex. And within that, there are a whole bunch of different proteins that are needed, any one of which could be the rate limiting step. The way I describe rate limiting steps, I always like to describe, if you go to a county fair, there'll be these food booths. And a food booth will be run by a community group, the soccer team. And you can watch them working in there, and none of them have ever worked in a kitchen before, And there are several steps. They're making a cheeseburger. You've got to order a cheeseburger, and they're going to make it. And you can just watch them. And there's a long line of people waiting to get cheeseburgers. Why are they waiting? And the answer is there's a rate limiting step in cheeseburger production. And let's just say it's the person who's putting on the pickles. This pickle lady is really chatty. And you can just watch her in there, right? And it would be useless to say, hey, can't you guys make burgers faster? Can't you cook them faster? Because you could cook them faster, and that's not the rate limiting step. The only way to make cheeseburgers faster is to tell her to get to work. And that's what's going on here, is that there's a rate limiting step, and the transcription factor is targeting and regulating that. And that's affecting the rate, like that. And that's always what we're talking about in this, is affecting the rate limiting step. And sometimes it's something in the pre-initiation complex. Okay? So that's the first one. And we just need to get, the transcription factors and the RNA polymerase all set, all good to go, like that. Okay? So then the next next example is that sometimes that's all good to go. Things are, that's not the problem. Sometimes the problem is that this DNA is all folded up and packed away. So the DNA can be wrapped around proteins that are called nucleosomes. and the DNA is just folded away like you folded a winter blanket in the spring, and you're like, we'll put that away, we're not going to use it. And the DNA plus these proteins is called chromatin. And we need to unfold this chromatin somehow. We need to unravel it, and that's the rate limiting step. Is we got to get the blankets out, shake them out, get them on the bed, like that. And we call that remodeled chromatin because there are lots of different ways that it can be folded away. And so sometimes that's the rate limiting step. It's not the pre-initiation complex. In those cases, the transcription factor targets the chromatin and changes the folding of the chromatin, like that. And there's some really famous transcription factors that do that. We'll learn about a couple examples of that one. And then so that's our second thing, is we need to get that chromatin unfolded. And the transcription factor targets that story. And then the third one is this idea of a pause polymerase. And it's the RNA polymerase. And what happens there is the rate limiting step is later. The pre-initiation complex, all set. The RNA polymerase binds, ready to go. The chromatin is unfolded. The RNA polymerase starts to go, and it goes for, it's about 70 nucleotides, and then it just stops. And it pauses or it arrests. And that's the rate limiting step. The analogy with the food stand is like, imagine the rate limiting step is the person who's actually handing the cheeseburgers to the customers, and they just don't let go of the plate. They're like, do you see the tractor pull? It was so good last night. And the people are tugging at their plate going, yeah, I'm hungry, I gotta go, right? And the person's just hanging on. It's the weirdest rate limiting step where you're like, oh my God, right? Not the pickle lady. She's just hanging out, nothing to do, because there's just a huge backlog of burgers, because the person who's at the very last step is actually coming out of the booth and being like, hey, wait a second. Oh, okay, you good? Got mayonnaise? And then they come back and they get another one and they're like, hey, buddy, what's that? Where'd you get that shirt? Like that. This arrested polymerase is the weirdest one because it's like the last possible step, like that. Way, way late. MYC is regulated with arrested polymerase. That's one way. We said it's regulated on this elongation. And Joe's question about elongation, this is a good place for us to focus on just a little bit of learning about this elongation. And the elongation is, in some cases, the rate limiting step. And the transcription factor needs to get it going. And we'll learn of an example of a transcription factor that targets that. It says, go, you arrested polymerase.
So with the MYC step, if it's relating to the MYC regulation, it's kind of like that person who betrayed, it's like, oh wait, this burger might be poisoned. So they're like
yeah, I don't really want to let go of this. It's pretty dangerous. Yeah, we should talk about it a little bit, like that. Yep. And the transcription factor helps get it over that hump. And so those are the three that we want to learn about. And for a different specific gene, it's a different rate limiting step. And so we'll learn about a couple of examples. Yep.
So with the RNA polymerase, it's like, I don't understand like where it's paused.
Yeah. So it's like, you imagine you're like, hey, you guys, you want to go to the mall? It's open. Come on, let's go. And you're like, wait, what? And then you're like, I should get my stuff together. Okay, we go out, we get in the car, and we just drive 100 feet, and then we stop. That's what it's like. It's like, oh my God, I thought we were going to the mall, and I'm like, we'll get there. Yeah.
It just doesn't finish its job.
Yeah, it just starts it, and it's like in the car on the way, and you're like, oh my God, I can't believe that the light turned green, and we're just paused here. You know, we have a goal. You know, we talked about it. We always stopped together. We got money, hats, you know. But here we are, stuck at, just paused. Yep.
Is that because like a transcription factor just like goes away or something? Like
yeah, that it needs a transcription factor to push it past that.
Okay.
And that's the rate limiting step, right? Everything else has happened, gotten done, and we're partly done with the task, but we just pause and just wait. And we wait until a transcription factor says, go, come on.
So it's like normal for this maybe.
Yep.
we
Yeah.
have this
Yes.
class,
Yes.
it's required,
And
it's normal.
so, yes. And the people who study this say that that's surprising. But 70% of human genes are regulated at that step. Weird thing. You know, it's just a weird thing, unexpected. But it's just a very late rate limiting step. Okay? So we're going to talk about that. That's a kind of another kind of way of driving a core piece through chapter 9 is understanding the role of the transcription factor and the different things. And our book does talk about this, but our book adds a lot. When it talks about transcriptional elongation, it's like, right? For us, it's like, well, it can be a rate limiting step in the expression of some genes, and so it would be targeted by the transcription factor. And we'll pick a transcription factor and learn about it and be like, oh, that's an example of a transcription factor that affects arrested polymerase. Okay? So let us pause here and have a concept chat and try to talk to somebody different each time, you know, move around the class, say hi, and we'll just get this done. What are 3 important things we talked about in class today? So far. So we'll do that, and we'll just have a good old day.
Do somebody different. Sorry, Joe.
Oh, and I didn't hand out the sign-up sheet yet, so let me get that organized. Yes, Godwin.
Super enhancer, what's that? Oh,
Yeah, it just means like really strong. So like it would be really activating it. So like if it was a rested plasma it would just be going, that transcription would be like, go, go, go.
oh.
Really strong. Yeah?
So it's
Yeah.
not different, it's just faster.
Yeah, it just means really strong, really powerful. Yep. Hi.
Do you have this sign up?
I do. Yes. Thank you, Tiva. Yep. All right, we're handing out the sign-up sheet. Kind of late here today, folks.
That's honestly.
Yeah, fair. Okay.
Do, 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 do.
So it's made too much meat.
It's like those 3 ways that transcription will regulate super controlled.
Nicholas.
Yeah. It's dangerous.
That's overexpressed.
Hi, I figured that
I mean, I know exactly.
We'll just do this another minute. all right, Hannah, what is one thing that you or our group is talking about that you thought was important today?
We talked about how MEC doesn't have the mutations necessarily in the coding region, instead it has in the regulatory region compared to RAS.
Yep.
And I think, that's a really important aspect that, when you have these mutations in these different regions is really like the outcome. It's
Right.
really constant.
Yep. Yeah. And so always this stuff is physical, and if it got explained well, it makes sense. RAS makes a defective protein, the mutations must be in the coding region. MYC makes a normal protein, but it makes too much, and it overwhelms all of the, all those protections by just making too much. Those mutations, those problems must be in the regulatory region. Yep. Eva, what's one thing you guys talked about?
Talk about that. We also found it interesting how, like with a PCR, how using a reverse transcriptase is become a really good tool in terms of researching.
Yep.
DNA.
Yeah, so, we're still kind of talking some about chapter 4, and that's called the fundamentals of molecular biology, and the emphasis there are, there's a list of tools that I said, these are important, and the book does a good job explaining them. It does a good job explaining reverse transcriptase. I wanted to talk about a couple to sort of showcase this reality that molecular biologists just love, that they can make discoveries in biology and then be like, oh my gosh, you could use this, we could sell this, like in a freaking vending machine, right? It's just nutty to see that, right? And then, Zach.
I'll take a shot at the three major ways transcription factors regulate transcription. Can I use your metaphor, like the burger analogy?
Burger one, or some metaphor. I mean, it's really interesting because you've learned about rate limiting steps, and they're super true. Let me just insert this. So, sometimes you're in a traffic jam on the highway where there's multiple lanes, and I've seen people, friends of mine, posting on social media this idea of, so there are going to be 2 lanes of cars waiting for this accident ahead. And what about it when there's one long lane that's full of cars, and then some people kind of just keep scooting up in that other lane and join in? So some people say this in social media. They say, I think that's actually good because we're getting cars lined up, and we'll say, we're using up the unused road space to get them all lined up really well, like that, okay? Like that, and that's helpful, right? It's not. Because the rate limiting step is at the point of the accident thing, and it has nothing to do with the cars ahead of it. Imagine this, right? Imagine this. The rate limiting step is the pickle lady. On the table in front of her are 40 burgers waiting for her to add pickles, waiting. Those are cars waiting, right? Does it matter how we arrange those burgers? It could be 1 line of 40 or 2 lines of 20. Is that going to help? No, because it's always about the rate limiting step. The rate limiting step is freakishly significant. It doesn't matter if you alter another step, whether it's a metabolic pathway or transcription regulation. And in transcription regulation, in our clay model, it was pretty simple. But it turns out there are several things happening, and one of them is going to be rate limiting, like by definition. And if you want to regulate something, you have to regulate that. But anyway, you're back to that analogy. Yep?
Even if everything was like that, it wouldn't matter. But, all right, well, the pre-initiation, that's the rate limiting step. That's actually, is that the cheeseburger?
It can be, and it can be this thing where you're like, oh my God, we have the various condiments out for the pre-initiation complex. We have a jar of mayonnaise and mustard, but no ketchup. That's slowing us down. We need to get ketchup out here to be ready.
Well, and then
all the stuff.
For number 2, the remodeled chromatin, which I'll just say somebody just put all the ingredients all together on one thing, and everyone's fishing off it to try that.
Yeah.
And then 3 would be if the person handing you the burger was, say, holding on to that thing, they're not letting go yet because that thing might be bad. Which in that case, I wrote, actually for Mick, that's the poison burger. the release, the RNA polymerase, it's just that annoying person that's holding the weight.
Mm-hmm. Yep. Yep. And so, for Mick, one of the issues is that getting that arrested polymerase, that paused polymerase to go, and that's called elongation, to elongate the RNA. So these strong regulatory regions, like in the retrovirus, they're just like, go now, as fast as you can, like that. And for these really strong regulatory regions, the transcription factors are multiple, and they are going to activate the rate limiting step, pause polymerase. And then if there's another secondary rate limiting step, they'll activate that. And if there's another one, so there'll just be, it's just like burgers, like that. And that's true for these really strong regulatory regions, and they just overwhelm whatever arrested polymerase and stability and stuff like that there are. They just make a ton. Yep. All right. Great. So we're doing great, and we're out of time. But on Thursday we'll talk about this, and we'll use this as a way of organizing lots of Chapter 9. So you want to bring questions from Chapter 9, and we can clarify them, and then. Next Tuesday, a week from today, we'll definitely talk about Chapter 17, of which we're already somewhat expert. All right? Great. So the sign-up sheet just came around now, so find that. Joe's holding the sign-up sheet, and that's that. And now the sign-up sheet is up here. Thank you.